Well, welcome everybody to today's online edition of the VCCC's Monday Lunch live stream. I'm Gavin Wright. I'm the Research and Education Lead for Lung Cancer for the VCCC, and a thoracic surgeon and a colleague for quite a long time of today's speaker. I'd first like to acknowledge the traditional custodians of the lands on which I and today's audience are located, wherever you may be. I'd also like to pay respect to elders, both past and present, and extend that respect to other Indigenous Australians present online. There are many interesting speakers coming up on Monday Lunch Live, so make sure you register your interest each week on the website. And we also have a learning page if you would like to view the previous topic. So there's the series, just the next 4 coming up. So about today's, if you want to ask a question, there is a Q&A and a chat button at the bottom of your Zoom screen, same as any normal Zoom meeting. So if you would like to, just put in a question there and we can endeavour to answer that for you. Today's presenter is going to talk about targeting ALK in lung cancer from discovery through to practice-changing clinical trials. Our speaker, we're very lucky to have Professor Ben Solomon, a medical oncologist in the lung service at MacCallum Cancer Centre. He's the group leader of the Molecular Therapeutics and Biomarkers Laboratory in the Research Division. His research focuses on clinical and translational research in lung cancer and the development of novel anti-cancer therapies. He's been involved in the clinical development of novel targeted therapies in lung cancer, including inhibitors of ALK, ROS1, RET, and MET. He's had a leadership role in the phase 1 to 3 clinical trials that led to approval of the ALK inhibitors crizotinib and Lorlatinib, which have changed many people's lives. I'm proud to be a long-standing colleague of Ben's. I'm very happy to hear from him today on a very topical month for us. Thank you, Ben.
Thanks very much, Gavin, for the kind introduction. Let me just share my slides. Yeah, thanks, Gavin. And as you mentioned, it's a great privilege to be able to speak to you all today, and in particular because November is Lung Cancer Awareness Month. So I just wanted to start with just a few little facts and numbers about lung cancer. Lung cancer is the most commonly diagnosed cancer anywhere around the world, with nearly 2.1 million new cases diagnosed each year. And it accounts for more cancer-related deaths than any other cancer type. It accounts for close to 20% of all cancer deaths. That's 1.8 million deaths worldwide and just over 9,000 deaths in Australia. Now, this is the image that most people have as an association with lung cancer, that of smoking, which is a major risk factor for lung cancer. But we know that there are many faces of lung cancer, and lung cancer is frequently diagnosed in people who have never smoked, such as Lillian and Alexandra shown here. In fact, in Australia, about 15% of people diagnosed with lung cancer have never smoked, and many more have quit smoking, often many years before their diagnosis of lung cancer. In women, one in 5 women diagnosed with lung cancer in Australia will never have smoked prior to their diagnosis. And in some parts of the world, like East Asia, up to 50% of women diagnosed with lung cancer have never smoked. And one of the consequences of the association of smoking with lung cancer is stigma. And this stigma has negative effects on patients in terms of delays in diagnosis, delays in accessing treatment, also impacts on patients' perceptions of themselves. And also it impacts on research funding. And here's our colleague, Peter Mack's very own Mary Duffy, telling us FFS to be free from stigma for lung cancer, which was a great campaign that the Lung Foundation led, fronted by our own Mary D. So why are outcomes so poor in lung cancer? It's in part because in most cases, lung cancer is diagnosed when it's advanced or metastatic. And outcomes fall, for example, with early stage disease, 5-year survival is above 50%. With local regional disease, 5-year survival is close to 30%. But with advanced disease, 5-year survival is only around 5%. But there is good news here. These are recent data from the AIHW, and that shows that in recent years, there's been a fall in cancer mortality in people diagnosed with lung cancer, or to say that this the other way around, there's been improving 5-year survival in patients diagnosed with lung cancer. And I hope to show you that in part this is related to the better treatments, including targeted therapies that we now can offer people with lung cancer. So in adenocarcinoma, which is the most common histological subtype of non-small cell lung cancer, it's possible to identify a potentially targetable oncogene in more than 50% of patients. In 2006, when I came back to Peter Mac from working overseas, only one of these targets, the EGF receptor, had drugs that were available to target it. And I'll talk to you about the progress made against another one of these targets, ALK gene rearrangements. But I should add that there's a whole range of other targets which are now actionable. So ALK, or Anaplastic large cell lymphoma kinase, takes its name from this unusual lymphoma called anaplastic large cell lymphoma. And there's a translocation between chromosome 2 and 5, which results in the fusion of nucleophosmin with a kinase, which subsequently became called ALK, which was identified in this tumor type. In 2007, a group of Japanese researchers identified ALK gene rearrangements in patients with non-small cell lung cancer. And it turns, and the most common fusion partner in lung cancer is a gene called EML4, although there are a number of other fusion partners, and there are also other variants. But what all of these have in common is that they result in abnormal expression and activity of the ALK kinase, which results in uncontrolled growth and proliferation of cells that have this gene. In other words, they're transforming and make the cells cancer cells. There are a number of features, clinical features of patients with tumors that have ALK rearrangements. The tumors are almost always adenocarcinoma histology. Patients are typically never smokers or have a light smoking history. And interestingly, they're often younger patients. The median age of patients with ALK rearrangements is about 50 years of age. They can be detected in tumors by various techniques, immunohistochemistry, FISH, or next generation sequencing. And as I'll show you, it's important to identify tumors with ALK rearrangements because it turns out that they're very sensitive to treatment with ALK inhibitors. In 2007, the same year ALK gene rearrangements were identified in lung cancer, we opened this trial at Peter Mac. It was a phase 1 trial with a drug called crizotinib, a small molecule inhibitor. It was actually designed as an inhibitor of c-Met, but it turned out that it also had activity against ALK and ROS1. And the trial began as a conventional phase 1, looking at escalating the dose of crizotinib to determine the optimal dose. But it was a little bit ahead of its time and used a design which is now actually very common with trials of new targeted agents, where following the phase 1, there was a part 2 where there were expanded cohorts in molecularly defined subsets. And following the identification of ALK rearrangements in non-small cell lung cancer, one of the major, a major focus of the trial was patients whose tumors had ALK gene rearrangements. So what we saw in the phase 1 study is that the majority of patients, or essentially almost all patients who had ALK rearranged lung cancer who had treatment with crizotinib, had some degree of shrinkage of their tumors with crizotinib. And this led to an objective response rate by RECIST criteria of 61%. The responses were pretty dramatic. Patients developed rapid improvement of symptoms and very early changes on scans, such as in the PET scans shown here. A second study, a phase 2 study, confirmed the findings of the phase 1 study and led to very rapid approval by the FDA in August of 2011. So crizotinib was approved in the US for lung cancer patients on the basis of around 250 patients treated with drug. Unfortunately, around the rest of the world, additional evidence was needed in terms of randomized phase 3 studies comparing crizotinib to existing standards of care. So the previous upfront treatment for newly diagnosed lung cancer patients was treatment with chemotherapy with this combination of platinum and pemetrexed. So we did a phase 3 study called Profile 1014, which compared crizotinib to pemetrexed as the initial treatment for patients with newly diagnosed ALK rearranged non-small cell lung cancer. And the study allowed crossover to crizotinib on progression after chemotherapy. And the primary endpoint of the study was progression-free survival by independent review. And the study was strongly positive in favor of crizotinib. The response rate was higher with crizotinib than with chemotherapy. The hazard ratio for progression-free survival was 0.45. So that means that patients had their disease controlled for, had twice the chance of having their disease controlled with crizotinib than chemotherapy. And the median progression-free survival with crizotinib was around 11 months. This led to the approval and reimbursement of crizotinib in 2014 and established initial treatment with an ALK inhibitor as standard of care for newly diagnosed patients with ALK rearranged lung cancer. We also learned, so this is the overall survival curves in that study. Now, it's important to note that patients treated with chemotherapy were able to cross over to crizotinib, and the vast majority of patients did cross over. And this is why I think the difference in survival curves doesn't reach statistical significance. But I guess the point that I wanted to make was prior to these drugs being available, the expected survival of a patient with an ALK rearranged lung cancer was no different to a patient with what we call wild-type lung cancer, or for whom no mutation was identified. And this was a median that was a bit less than 1 year. So this showed that the survival of these drugs really do alter the natural history of the disease. And the median survival had not been reached after 5 years of follow-up. We know, however, that patients do progress on crizotinib, as you saw from those curves. And this can be, this often happens in the brain, but can also happen outside the brain due to what we call acquired resistance. And newer molecules have been developed, so-called second and third generation inhibitors. And what they have in common is they're all more potent against ALK than crizotinib. They cover mutations to differing degrees that confer resistance to crizotinib, and they tend to be better at getting into the brain. One of these is a drug called alectinib, and alectinib was compared to crizotinib in a phase 3 study called the ALK study. And the study again was positive, with a hazard ratio of again 0.47, this time of alectinib over crizotinib, again indicating the sort of rapid progress and improvements in outcome with these newer drugs. The study was recently updated, and the median progression-free survival with this drug was 34 months. Compared to about 11 months with crizotinib. And we also have updated overall survival with this study. And at 5 years, more than 60% of patients were still alive on the alectinib arm. So with these new drugs, the algorithms for treating ALK-positive non-small cell lung cancer have changed from an initial first-generation inhibitor, then followed by a second-generation inhibitor, to upfront use of a second-generation inhibitor. But then it remains what to do in the context of progression. And progression can occur due to multiple reasons, and how it's managed depends on a number of factors. If it's what we call oligoprogression, if it happens in only one site, it might be possible to use a treatment such as stereotactic radiotherapy to do it, to treat that site and continue on the drug. But the major mechanisms of resistance we see are failure in the brain, or progression due to acquired resistance in the tumor. And this can happen due to the acquisition of ALK-dependent resistance, which, as I'll show you, is usually due to ALK mutations, due to activation of bypass pathways around ALK, or to histological transformation. So in the context of acquired resistance to crizotinib, mutations that occurred in the ALK kinase itself were identified. These can occur at a number of different sites and interfere with the ability of crizotinib to bind and interfere the ALK kinase domain. And this can also occur in the context of amplification of ALK as well. When you look at resistance occurring in the context of second-generation drugs, such as ceritinib, alectinib, or brigatinib, it turns out that these mutations are more common than after resistance to crizotinib. And a particular mutation, the G1202R solvent front mutation, tends to be the most common mutation identified. So lorlatinib is a novel third-generation ALK tyrosine kinase inhibitor. It's got a very unique structure, this circular or macrocyclic structure, which enables the molecule to be very compact, and it's very, very effective at crossing the blood-brain barrier. And in terms of its activity against ALK, it turns out that Lorlatinib is the most potent ALK inhibitor against wild-type ALK, but has a very broad spectrum of coverage of mutations, including activity against the G1202R mutation, which confers resistance to the other first and second generation ALK inhibitors. And this is a preclinical study showing evidence of penetration of Lorlatinib into the brain. So on the basis of its ability to penetrate into the brain and its potency and spectrum of coverage, it was selected for clinical development. And we were involved in this study, the phase 1/2 study with Lorlatinib, here at Peter Mac, and the study started in April of 2014. And the study had a complex design. There was a phase 1 part, and then an expansion with multiple different cohorts depending on prior treatments, and also ROS1 cohort. And on the basis of activity seen in this study in several of these cohorts in patients who had received prior second generation inhibitors, Lorlatinib received approval in the US and around the world, including Australia, for this indication. And I'll show you some of the data for that. So these are data from a cohort of patients who had received 2 or 3 prior lines of ALK tyrosine kinase inhibitors, with or without chemotherapy. So a very heavily pre-treated cohort of patients. And in this population, the confirmed objective response rate was 38.7%, with a median progression-free survival of 6.9 months. And activity was seen in the brain with an objective response rate of 53%. And it's important the brain activity is really significant because there is a cumulative incidence of brain metastases. And in this study, depending on the cohort, up to 50 or even 80% of patients had brain metastases on entry into the study. And this led to FDA approval in 2018 and TGA approval and PBS listing in 2020. So the gap that you saw with crizotinib of nearly 3 or 4 years does seem to be narrowing with Australian drug approvals. We looked at the profile of mutations within tumours, both by tumour biopsy and by liquid biopsy, looking at plasma circulating tumour DNA. And as expected, the G1202R mutation shown in blue was the most commonly identified mutation, and responses were seen in this type of mutation, but also across a range of other mutations. And in this G1202R population, the drug was clearly active, as seen in this waterfall plot here. What was interesting was differences in response rates in patients whose tumours had mutations identified compared to patients who didn't. So this is responses in patients with mutations biopsied on progression after prior therapies by tissue. The blue bars and curve are patients who had mutations identified. And here the response rate was 69% and the progression-free survival around 11 months. The response rates and progression-free survival were less in patients who didn't have mutations identified, but nonetheless, as you can see from the red bars, there still were patients who responded and benefited from treatment. When you look with plasma, which is a much easier sample to obtain, the difference is in the same direction, but of a lesser magnitude. And this probably reflects, to some degree, false negatives or the difficulties in identifying mutations in plasma. But again, benefit in both patients with and without mutations. I also want to draw attention to the activity in the brain. And as you can see from the waterfall plots, the activity in the brain was pretty significant, including quite frequent complete responses. And I was going to illustrate this with a case history from a patient who actually didn't make it onto the study. So this is a young man who was diagnosed in 2014 with advanced ALK positive non-small cell lung cancer, whose disease had metastasized to within his lungs, to the lymph nodes in his chest and into his brain. He commenced treatment with first-line Lorlatinib, a second-generation ALK inhibitor, in November of 2014, and had a good response to treatment. However, 2 years later, he had progressing disease both in his brain and chest and was referred for consideration of treatment on the study, which I mentioned. Unfortunately, the disease in the brain progressed very rapidly, and he presented with seizures to the emergency department and had a scan, which you can see on the left there with, you can see 3 large masses on the left side of his brain with surrounding edema and what we call mass effect. So he presented to the Royal Melbourne Emergency Department. He had urgent surgery with resection of that middle tumour that was causing the mass effect. And he commenced treatment within a week of his surgery with Lorlatinib outside the study because he wasn't eligible for the study. Incidentally, he made a pretty quick recovery from his surgery, and this was not long after we'd moved into this building, the then new VCCC building, and got married on the rooftop, the 7th floor rooftop of the building whilst an inpatient. He, 4 years later, remains well on Lorlatinib with a complete intracranial and extracranial response. So in the MRI shown here, you can see the cavity from the resection, but no other disease. And I was very delighted last week to get a photo that he sent from a hike he was doing in the mountains in Tasmania. And this is about 6 years after his original diagnosis with lung cancer metastatic to the brain. So this is Lorlatinib in patients who had progressed on prior inhibitors. What about using Lorlatinib as an initial treatment? There was a cohort in the phase 2 study of patients who had not had prior treatment. Just a cohort of 30 patients, but they seem to have a quite high response rate, a response rate of 90%, and a median progression-free survival that had not been reported. So on this basis, we went on to do a phase 3 study in newly diagnosed patients with ALK rearranged lung cancer, comparing treatment with upfront Lorlatinib to Crizotinib, which was standard of care at the time the study was initiated. Although, as you've seen earlier, Alectinib is probably now the standard of care. The primary endpoint of the study was progression-free survival by blinded independent central review, and there were multiple other secondary endpoints which are shown on the right. So this is the primary endpoint of the study. The study was strongly positive for the primary endpoint of progression-free survival, with a hazard ratio of 0.28. This means a 72% reduced chance of progression with Lorlatinib compared to crizotinib. And it's difficult to make comparisons across studies, but the hazard ratio with second generation inhibitors such as alectinib and brigatinib tends to be in the order of 0.7 to 0.50, suggesting that this is the most potent or the most effective drug reported so far. And the response rate was 76% with Lorlatinib compared to 58% with crizotinib. Benefit was seen in all subgroups that were looked at. And in patients with brain metastases, the hazard ratio was 0.20. These are responses in the brain, both in patients with measurable disease and non-measurable disease. It's probably easiest to focus on the patients with measurable disease at baseline on the right. And this was assessment with MRI scans and independent neuroradiology review. And the response rate was 82% with Lorlatinib compared to 23% with crizotinib. And the intracranial complete response rate was 71%. So really super effective in the brain. This plot shows intracranial time to progression again by MRI scan. And what this shows is if there's progression in an existing brain met or development of a new brain metastases. And as you can see, there were very few progression events in the Lorlatinib arm, which almost looks like a straight line, whereas there was steady progression in the brain on crizotinib. And this speaks to the importance of having drugs that effectively target the brain, and also speaks to the ability of Lorlatinib not only to delay progression of existing brain metastases, but potentially to prevent new brain metastases. Overall survival is not yet mature, with fewer than 20% survival events, so very early in the study. So algorithm for treating ALK positive lung cancer continues to change. On the basis of the trials that have been done, we now have the options for third generation inhibitors after progression on second generation inhibitors, and potentially might soon have the option for a third generation inhibitor first. And I think I started off with a slide showing improved survival in patients with lung cancer. And I think the story of ALK inhibitors in the last just over 10 years really does speak to the progress that has been made in this area. We knew that prior to ALK inhibitors, survival in ALK positive lung cancer patients was very similar to patients without ALK rearrangements, around a year. But there's been steady progress with the ALK inhibitors, and with newer generation inhibitors, we can expect median survivals in excess of 5 years, which I think is significant progress, but also speaks to the fact of us needing to go much further, given that many of these patients are young patients, often with young children, and 5 years is a good start, but indicates a long way to go. And I was delighted in a recent lung clinic to have a patient bring a cake, and the cake was to mark his 10-year anniversary of living with advanced lung cancer. And he was one of the patients who was enrolled on the crizotinib phase 2 study. And we're seeing more and more, not necessarily cakes, but patients getting to significant anniversaries like this with new treatments. So how can we extend these gains? I think one really important way is to move these drugs into early stage disease. We've recently seen that with EGFR inhibitors, and this is a study which we're currently doing called the ALENA study, where patients with resected ALK-positive lung cancer are randomized to treatment with Lorlatinib for 2 years or to chemotherapy. And we don't know yet whether this study will be positive or not, but potentially this might be a way to potentially result in cures from drugs like Lorlatinib, which is ultimately what we would like to do. So in conclusion, ALK rearrangements are found in about 3 to 5% of lung cancers and confer sensitivity to ALK tyrosine kinase inhibitors. As I mentioned, we've come a long way. We've had clinical trials that have led to approvals of first, second, and now third generation ALK inhibitors. And I think these have really transformed outcomes for patients with ALK rearranged lung cancer. But we still have a way to go, and we still don't know what the optimal sequence of ALK inhibitors is, how to choose between drugs, and potentially how to partner ALK inhibitors with other drugs to prevent or delay resistance. And hopefully these studies that address these questions will continue to improve outcomes. And finally, I'd like to thank all the patients and their families that have been involved in the clinical trials that have really transformed outcomes in this area, and to our amazing Peter Mac lung cancer clinical trials team, which have been involved in the trials way back from 2007. And thank you all for your attention.
Thanks very much, Ben, for an amazing talk. What I'm going to do now is we'll go through a few questions that have been asked throughout your talk. And I'll start with a quick one myself. Is Alectinib or Lorlatinib available first line for patients given or is that still in a trial setting? Are they PBS approved?
Yeah, so Alectinib certainly is available first line. So we've actually got 5 drugs that are PBS approved: crizotinib, Alectinib, Brigatinib, Ceritinib, and Lorlatinib. It sounds like almost too many, but in terms of first-line treatments, Alectinib and Brigatinib are probably our go-to drugs. Lorlatinib at the moment, so those results that I showed are pretty hot off the press. We just presented them about a month ago at ESMO, and they're yet to come out in press, but hopefully that will result in Lorlatinib being an option for first-line treatment. Currently, it's only available on progression after a second-generation inhibitor.
Sure. All right, our first question was, what's the difference in chemical property of Lorlatinib that allows it to remain an effective therapy against many of the common pan-resistant mechanisms that other ALK inhibitors could not target?
I think it's some really clever chemistry. It's the shape of the molecule, which is really quite different to the other compounds. It's got this circular structure, which makes the molecule really very compact. And not only is that a good thing for penetration into the brain, but it also means that there's less ability for steric interference from the mutations with Lorlatinib binding to its active site, or the site it needs to be active against ALK.
So it really gets in like ink into the chalk. Is that right?
If you remember your Colgate ads, Gav, yeah.
Next question was, in the Lorlatinib study, did you use next generation sequencing to determine if there were mutations in the ALK?
Yeah, so the mutations I showed were either from tumor tissue or plasma. The mutations from tissue were by next generation sequencing, and the plasma was by also a next generation sequencing, a commercial platform called Garden 360.
Right, I have a question from one of the more famous ALK patients, Lisa Briggs. She's asked, with such transformative changes within the landscape of lung cancer for patients, both through survival benefits and treatment options, what do you see as the potential future for treatments beyond Lorlatinib?
Yeah, no, that's a great question, Lisa. I think we've got great drugs. I think the areas that sort of need exploring are combinations, and also about how we sequence them and whether we can use real-time assessments such as from plasma to guide switches before resistance emerges. So if we can see from a plasma test an evolving mechanism of resistance, and if we can cut that off at the pass, that might be a way to extend things further. And as good as Lorlatinib is, tumours will also develop resistance to Lorlatinib. And I think it'll be, again, important to sort of understand how that happens, ways to deal with it, and also ways to prevent that resistance from happening in the first place.
We've got a few more minutes for questions. If anyone wants to use the Q&A button at the bottom of the screen. In the meantime, I'll just ask another question myself. It's obviously, this is just fantastic that ALK fusion lung cancers have got these treatment options. Do you see these spreading to, do you think there'll be other lung cancers or other adenocarcinomas that may see developments in inhibitors? And my other question related to that would be, is there anything on the horizon for the now sort of forgotten squamous cell cancer, which probably still makes up 20 or 30% of our lung cancers?
Yeah, I think the paradigm of, I guess it sort of started with EGFR and has certainly extended to ALK, will definitely apply to other rarer molecular subtypes. We've seen this year, we've seen approvals for the first time of potent selective RET inhibitors, potent selective c-MET inhibitors, and it's likely that there'll be newer generation compounds that improve on those. A really exciting advance is inhibitors that target KRAS mutations, in particular G12C, which is about 50% of KRAS mutations. Squamous cell carcinoma tends to be much more tricky from the perspective of targeted therapy, but I think the immunotherapies hold a lot of promise in that particular context.
Got another question from Ali B. After alectinib with CNS progression, what's the next best drug?
So after alectinib with CNS progression, so alectinib has good CNS penetration, but we've in the phase 1/2 study, but also outside the phase 1/2 study, we've seen good responses to Lorlatinib after progression on alectinib in the brain. Again, just speaking to its structure and how it's able to get into the brain. And it's also active against leptomeningeal disease, which is a really tough to treat situation.
Yes, it's certainly exciting, the KRAS. For the audience, KRAS mutations are very common in lung cancer. So to have something that treats that group is is an amazing development. Can you just speak a little bit more on how you see the promise for the KRAS inhibitors?
Yeah, so there are a whole bunch of KRAS inhibitors in development. The first of those is from Amgen. But interestingly, the response rates to KRAS, to the G12C, and they only work against a particular mutation, this G12C mutation. The response rates seem a bit lower than what we've become used to with EGFR and ALK, for instance. The response rate seems to be around 30, 35%. The reasons for that aren't clear. It's not clear whether it's because these first generation drugs aren't potent enough, or whether there's some biology in the context of RAS mutations that no matter how good or potent your drug is, that's going to be the sort of range of response that you're going to get. But there are a whole heap of newer RAS inhibitors, and there's early moves into combinations, I think, which will hopefully lift that response rate.
I've got a question from Grace Chazan. What factors do you and the MDM use to decide about local therapy for brain mets versus immediately starting Lorlatinib on CNS progression on earlier generation ALK TKIs? How quickly does Lorlatinib work in the brain?
Yeah, it's a good question. I think so it can work very quickly, and you saw the data from the phase 3 trial with a very high complete response rate. I think the answer to that's changing. We're probably doing it a bit less than we would have previously, but if there is a single lesion that's suitable for stereotactic radiotherapy, I think we would still entertain using the radiotherapy to deal with that MET and change to a more effective or more brain penetrant drug. But there was a time, for example, when we just had crizotinib and no other drugs to move on to, that we would really utilize local treatments to a great effect and then continue beyond progression. But now we don't necessarily need to continue beyond progression when we can move to a more potent drug afterwards. But it's always it's not a straightforward discussion. It needs a multidisciplinary discussion and close discussion with the neuroradiation oncologist.
Got another question from Lisa Briggs. Can you indicate what the response rate is with the ALK trial, which we understand is now open at Peter Mac? I don't know whether she means response to the therapy or how well you're recruiting, but maybe tell us a bit about that.
Yeah, so thanks, Lisa, again for that question. So the ALK study is a study which is driven by Mal Itchens, who's a fantastic young oncologist from Sydney. And the study involves alternating treatment with Lorlatinib and crizotinib. Again, one of these ways to sort of trick the tumour into extending or delaying progression. It's a fantastic concept. It's still pretty early in the study, so we can't really speak to the response rate. But I think what will be key not so much the response rate, because patients get treated with Lorlatinib upfront for a few cycles, so the response rate will be pretty much what you expect with Lorlatinib. But it'll be how long the disease stays controlled for, and we're a long way off those data yet. And yes, the study has we were impacted by COVID like a lot of other studies, but we've sort of coming out of that and ready to roll with alkanate.
So it's really about putting off the resistance as long as possible and extending the valuable life of the drug.
Exactly. Yep.
Yeah. All right. Well, I think we've had an amazing talk there, really, of the entire life of ALK inhibitors from inception through to amazing developments and impact for survival and quality of life of survival of patients today that we're enjoying today. And I'd like to take this opportunity to thank Ben, who, if you didn't know, is one of the world's renowned experts on ALK, and thank him very much for today's amazing talk. And so I'll put my hands together for everyone online. And I'd just like to remind everyone again of the upcoming talks to come to as today's. They're all incredible talks that we're very privileged to be able to access. Thank you.